Good morning, I'm Elise Modica, and here's the latest update from Spectrum Autism Research. Excess of "don't eat me" cell signals may drive brain enlargement in autism, by Peter Hess. A "don't eat me" cell signal called CD47 contributes to brain overgrowth in people with idiopathic autism, according to unpublished research presented last week at Neuroscience 2022 in San Diego, California. And two cancer-linked cell signaling pathways may also be disrupted in people with above-average brain size, according to the new findings. Overexpression of CD47, which prevents immune cells called microglia from pruning neurons during development, leads to overgrowth in neural progenitor cells derived from people with macrocephaly and an autism-linked deletion in the chromosomal region 16p11.2, the same lab showed last year. The cultured cells also overexpress calreticulin, an eat-me signal, but CD47 overpowers it. Blocking CD47 corrects the imbalance and curbs neuron overgrowth in cultured human cells and in mice, the team showed. The new studies extend the 16p11.2 findings to people whose autism has no known genetic cause and identify genes and additional cell signals involved in brain overgrowth. We are trying to better understand whether there are common mechanisms shared between genetic forms of autism with macrocephaly and idiopathic forms of autism with macrocephaly, says lead investigator Sundari Chetty, assistant professor of psychiatry at Harvard Medical School. In the new studies, Chetty and her team coaxed donated blood cells from autistic and non-autistic people into stem cells and then into immature neurons. The cells from 4 people with idiopathic autism and disproportionate megalencephaly, a brain size in the 98th percentile, all overexpress CD47, unlike those from 4 autistic and 4 non-autistic people with average brain size, according to unpublished findings presented on the 13th of November by Anna Sherbina, senior machine learning engineer at Insitro, a drug discovery and development company in South San Francisco, California. Sherbina worked on the study as a graduate student in Chetty's lab. The cells from the participants with idiopathic autism and megalencephaly also overexpress a similar protein, CD99, the pilot study shows. CD99 plays a role in acute myeloid leukemia, and doctors have killed tumor cells in leukemia patients by treating them with an anti-CD99 antibody. Genes in multiple immune signaling pathways driven by CD99 are upregulated in the cells, according to unpublished results Sherbina presented on the 16th of November, further linking cancer pathways and brain overgrowth. This analysis also showed a strong correlation between CD99 and pathways involving meiosis, the cell cycle, and cell proliferation, all of which are also involved in cancer. This is very interesting because it mirrors our earlier observation in 16p11.2 deletion work, where CD47 played a role in cancer as well as brain overgrowth, Sherbina says, and it appears that CD99 has a similar link as well. Another unpublished study presented on the 15th of November by Siwei Chen, a postdoctoral fellow in Chetty's lab, investigated how genes in the 16p11.2 region influence CD47 levels. Chen used CRISPR to knock out each of the 29 genes in the 16p11.2 region from control human-induced pluripotent stem cells and narrowed in on 3 that affect CD47, KCTD13, MAPK3, and ALDOA. A literature review suggested that loss of function in these genes disrupts CD47 and p53-linked processes, including the normal response to DNA damage. p53 is a tumor-suppressing protein that is also affected by some autism-linked genes. CD47 is upregulated, and p53 is downregulated in cells grown from people with macrocephaly and 16p11.2 deletion, or idiopathic autism, Chen found. And markers of DNA damage, which triggers cell death, are dramatically downregulated in both groups with macrocephaly compared with people with typical head size, she found. Chen hypothesizes that a reduced rate of natural cell death could also contribute to brain overgrowth. Further research is underway to confirm the degree to which individual genes in the 16p11.2 region play key roles in dysregulating the natural process of cell death, Chen says. That's all for today. Check back on Wednesday for more content from Spectrum Autism Research or go to spectrumnews.org.